All right. Well, we have two keynotes this morning, and it's a great pleasure to introduce Jim Bever, who will be giving the first one. Jim and his wife Peggy, who unfortunately is not here today, are the co-coordinators of research at the Perennial Agriculture Project field station outside of Lawrence, Kansas. Jim also happens to be a very prominent professor at the University of Kansas, but that's sort of secondary in his role in the world. But anyway, I asked Jim if he would be willing to talk about the soil microbiome as it relates to a transition to perennial polycultures in agriculture. Jim has done most of his research in natural systems and prairie restoration, understanding the role of mycorrhizal fungi and other organisms and how they interact. This conference hasn't had a huge emphasis on the soil microbiome, but it is definitely an area that is receiving a great deal of attention right now, and we know it matters, and looking forward to what Jim has to say on this topic. So, Jim.
Thanks, Tim. Yeah, it's been a real pleasure to be here, so it's a fun conference. Most of my work is on plant microbe interactions in the context of plant species coexistence, or how the strength of that interaction changes over time, and it's been mostly done in the tall grass prairie. So it's been fun to think about the role of the microbiome in perennial polyculture production, and this has been done with Tim and David and the rest of the Land Institute team, and also in collaboration with the work I'm presenting with Peggy, Guanzo, Liz, and Tom. So plants interact with tens of thousands of different kinds of microbes continuously. So there's many ways that these microbes can influence production. I'm going to focus on two big categories of microbes that have the biggest direct effects on plants. So there's pathogens. So here's Agrobacterium infecting a plant root. The bacterial pathogens, fungal pathogens. Here's a cleared root with the fungus stained blue. This is a oospore of oomycete of a Pythium pathogen. There's several fungal pathogens in there. So it's well established that these pathogens can have strong effects on yield. But plants also associate with beneficial microbes. This is a cleared root, this is a cortical cell, and this is an arbuscule of an arbuscular mycorrhizal fungus, which is an important mutualist, as well as nitrogen-fixing bacteria. This is a nodule of a clover. And so these can be important determinants of production as well. So conventional industrial agriculture has basically, in an effort to remove this, has simultaneously removed this. So through intensive application of pesticides, tillage, and fertilizer, conventional agriculture has minimized the microbiome. And then in breeding for yield in environments in which these practices are in place, breeding has then minimized the importance of the potential interactions with these microbiome. So that's true in corn. It's not completely successful. Pathogen buildup in corn is a major driver of the rotation of corn with soybeans throughout the Midwest. But even soybeans with their nitrogen-fixing symbiont don't really benefit that much from it. Farmers apply nitrogen to these fields. So we're going to ask, can we better manage the microbiome in perennial polycultures? And that better management would be by maximizing the benefit of mutualists and minimizing the costs of pathogens. And so we'll do this, we'll address this issue by looking at the way the microbiome interacts and behaves in the native system. And in this, we're following Wes's tradition of nature as measure, as how might this microbiome work in a perennial system, and then also look at the annual crops, and particularly in the context of what drives the overyielding in polyculture. So we'll first look at how mycorrhizal fungi mediate the dynamics of prairie plants. We'll look at preliminary tests in Land Institute candidate crops, and then talk about whether there's synergism between when you have multiple symbionts in the roots for legumes, and then talk about pathogens in polyculture. Okay, so mycorrhizal fungi, there is a very old association with plants. The oldest plant roots were interacting with, have infections with these fungus. So a corn root growing into the soil will have these soil fungi will penetrate the root and form these structurals, arbuscules, in the cortical cells, which are the site of exchange of resources, phosphorus mostly from the fungus, for carbohydrates for the plant. And then these fungi have hyphae, which go throughout the soil and increase the access of the plant to phosphorus, and thereby promote plant growth. So these fungi can have lots of other effects on plants. They can increase plant defense. They could weaken plant defense. They can, but a big thing they do is they also stabilize soil and reduce vulnerability to erosion. So they're a major player in ecosystem services of the soil. So there are lots of different kinds of mycorrhizal fungi. These are nine different species of mycorrhizal fungi represented as their asexually produced spores. They differ in their size and shape and color. And importantly, they differ in their ecology. They differ in their vulnerability to different disturbance, and they differ in their effects on plants. So I'm going to talk about the dynamics of this system in the context of following up on what Tim had talked about in succession, that the function of this soil microbiome is degraded by disturbance and then over succession time improves. And we're going to talk about how that works with mycorrhizal fungi in the prairie system. So prairie remnants, unplowed prairie is highly diverse, full of long-lived perennials. With conventional agriculture, that's removed. And then with abandonment of conventional agriculture, if nothing is done, then the land is typically colonized by non-native weedy plants. If you take the seeds from the prairie and throw it into this environment, people have been successful in restoring the dominance of natives, but mostly through low diversity plantings that are dominated by the grasses. So restoration practice fails to recover the original diversity of the tallgrass prairie. And that screening is not random. So it's particularly the late successional long-lived plant species like the rattlesnake master or the baptisia here that are unable to establish in this environment, in this post-agricultural environment. And we ask the extent to which this difficulty in establishing these late successional species is due to the changes in the mycorrhizal community. So when we look in the soil, we see strong differentiation of the mycorrhizal fungal community from the remnant state to this post-agricultural state. There's a suite of fungi that are lost with disturbance, and then there's another group of fungi that proliferate with disturbance. So these are weedy fungi that are very common in post-agricultural environments. So does this degradation of the mycorrhizal fungal community limit the success of these long-lived plants? So these are the plants, these plants that allocate to rapid growth and have high investment in these root systems instead of mycorrhizal fungi that proliferate in these post-agricultural environments. These long-lived, slow-growing plants that have greater allocation below ground are less successful. And so we're going to look at how these plants interact with the mycorrhizal fungi. And so we do that by taking a suite of plants along a gradient of life histories, and we plant them into pots that are filled with sterile soil or sterile soil that's inoculated with mycorrhizal fungi derived from the native system. And so the first experiment done like this is from Liz Koziol, and so here on this axis we're looking at the ratio of the size of the plant with mycorrhizal fungi over the ratio of the size of the plant in the sterile. So when this ratio is 1, then they don't benefit from mycorrhizal fungi at all. So the early successional plants don't need mycorrhizal fungi, they don't benefit from mycorrhizal fungi, while the later successional plants grow much larger with mycorrhizal fungi. So So for example, Allium cinuum is 20 times larger with mycorrhizal fungi than without. So in this experiment, we just used a mixture of species of mycorrhizal fungi. It also matters how they respond to the different species, to differences in fungal composition. So we repeated the experiment, this time inoculating individually with many different species of fungi. And so here we're representing this. This is the change in percentage change in the growth of these plants. And we have 6 early successional plants, 6 later successional plants. And then they're grouped. These bars represent the growth response to different species of mycorrhizal fungi. And so what you see, 2 things. First, you see that the later successional plants, again, respond more positively than the early successional plants. But also, for the later successional plants, it matters a lot what the identity of the fungus is. So Allium cinuum here does really well with lamellosum, but not so well with Cetrospora pellucida. It doesn't matter so much for bidens. It does equally well for all of them. It just doesn't care. In this case, it didn't benefit. It was negatively affected by all of the fungi. So then if we go back, so the late sessional plants are much more sensitive to the composition. So if we go back to what we observed here, are the late sessional plants more dependent on the mycorrhizal fungi that were dominant in the unplowed prairie? And so we test that by inoculating these late sessional plants into these in placing them into this post-agricultural environment. And we consistently find through many of these types of experiments that the establishment, survival, and growth of these late sessional plants are greatly improved when they have their symbionts, their native late sessional symbionts with them. So that's encouraging that inoculation could matter to perennial production. So how about the candidate perennial crops that the Land Institute is considering? We first looked at this with a greenhouse assay where we tested many different many candidate crops as well as the annual congeners. And so same approach, inoculated with the native mycorrhizal fungi. So we then went into test whether we see similar responses in field inoculation. So this is the Perennial Agriculture Farm at the Land Institute property in Lawrence, Kansas. So this field was conventional agriculture for many years, and then it was planted into smooth brome. This plot here is an experiment where we inoculated and tested the native species of native prairie plants and their responsiveness to mycorrhizal fungi. So we know that in this degraded mycorrhizal community, these late sessional native plants are responsive in the background of this mycorrhizal community. So this experiment was a test of these 8 species in their responsiveness to reintroduction of native mycorrhizal fungi. And so we did 8 densities. We introduced 8 densities of mycorrhizal fungi through just tilling it in before planting from 0 to a large amount of mycorrhizae, and generally found that the response to inoculation was strongly predicted by the observed response in the greenhouse. So here, Kernza wasn't responsive in the greenhouse. I should have commented that in that greenhouse experiment in particular, it was a really high P soil that we had used. So I don't think this is the final word on Kernza. In the field, we see a weak positive relationship. For survival, these are different in growth from this, but there's a lot of variability. It's not a very strong effect. So that's consistent with Kernza not being super dependent on mycorrhizal fungi. And that's different from the response of silphium or sorghum. This is sorghum seed mass, and this is the rosette size of silphium at the end of the first year. And so in both these cases, we see strong responses to the density of native mycorrhizal fungi, in both cases resulting about a 10% increase in production. And we see a stronger effect. So we didn't see any effect in annual sorghum. We see a stronger effect in alfalfa for survival and total production, with about a 20% increase in both of those. And with a similar pattern in curiclover and lupin didn't respond. Lupin is a legume that doesn't associate with mycorrhizal fungi. So this is encouraging that manipulation of the microbiome can improve production in perennial systems. So if we look at alfalfa and curiclover, they also associate with rhizobium. And so is to what extent are perennials more responsive to rhizobium, and to what extent does co-inoculation of good rhizobium with good mycorrhizal fungi generate synergistic benefits for growth of these perennial legumes? Because we might expect that mycorrhizal fungi are providing phosphorus, nitrogen-fixing bacteria are providing nitrogen. These are complementary resources. So having both of them functioning at a high level might generate increased benefits beyond each of them individually. So by synergism, we mean that the mycorrhizal fungi benefit this amount, rhizobia benefit this amount. So the expected, when you put them together, you should have a much bigger plant. But you might get even bigger, and this would be the synergistic benefit from co-inoculation with good mycorrhiza and good rhizobia. So we asked whether perennials are more likely to give these synergistic benefits than annuals, and we did this by surveying the published literature. And what we found is actually very striking. So for annuals, on average, they benefit from rhizobia, they benefit from mycorrhizal fungi about the same amount, but if you put them both together, there's not even the additive expectation. They're basically the same size here, again, with the two symbionts as with the one symbiont. Doesn't matter which one you give, they benefit, but not that much. But for the perennials, they benefit much more from rhizobia, much more from mycorrhizal fungi, and when you put the two together, they benefit even more. So they have, if you add this up, it's like 10 times bigger, with more than about 14 times, and with the synergistic benefit, they're 2 times bigger with both of them than they would be alone on average. Okay, so what we're finding is that in contrast to annuals, long-lived perennials depend on mycorrhizal fungi. They're more sensitive to mycorrhizal fungal composition, and they respond more positively to native mycorrhizal fungi in particular. They don't like post-industrial ag mycorrhizal fungi, is what it is. Yeah. So, and they also respond more positively to rhizobia, and they respond synergistically to the joint legumes, respond synergistically to the joint inoculation of good mycorrhizal fungi and rhizobia. And field inoculation can improve the production of some of the candidate perennial crops. So this looks good for the microbiome's influence. But what about pathogens? So if we go back to the prairie system and look at how pathogen defenses vary along with life history in the prairie system, we tested the effect of a generalist bacterial pathogen, this is Agrobacterium, and it infects composites in general. And so we looked across composites, and we found that in the fast-growing early successional composites, a greater proportion of their mass was taken up by the gall that's formed from Agrobacterium, while a lower portion of the mass for these later successional species was made up of the gall. So that's indicative of later successional species generally being better defended against generalist pathogens. But most of the big pathogens in agricultural systems are specialists, and are much more specialist than Agrobacterium. They're fungal, and these can generate differential impacts with accumulation on each of these hosts, and we can measure that by this metric called feedback. And so when we look at the measure of plant soil feedback, negative effects are indicative of accumulation of host-specific pathogens, and so we looked across the prairie system, across early and late successional species, and measured the net feedback on them. And we found that these early successional species had strong negative feedback. So within 1 year of association with that plant, this plant was doing worse with its the microbial community that resulted from that, and that's largely due to pathogen accumulation. And the later sectional species are less vulnerable to that. In this case, they did significantly better when they grow with their own soil than the others, and that's likely due to the mycorrhizal fungi. So over the long term, so we have faster accumulation of pathogens on these short-lived plants, but over the long term, pathogens will accumulate on these long-lived plants. We know that from trees. Trees are in one location will accumulate pathogens that will prevent their seedlings from establishing near them. And so given enough time, pathogens will accumulate even on the most resistant perennial crop. And so can mixed cropping suppress the accumulation of these host-specific pathogens? You'd expect that because these pathogens don't grow on the other species. So we tested that with this greenhouse mixture of these late sessional prairie plants. This is a legume, a morphokinescence, big blue stem, and a composite liatris. And we grew them in all possible combinations where there are 4 plants in a pot, and we grew them in all different variations of the composition of these 3 species in those pots. And the pots are filled with sterile soil, native mycorrhizal fungi, post-agricultural microbes, or native prairie microbes. And it was just grown for 1 growing season, and we measured the biomass of everything, and we looked at a couple other things in the process of that. And what we found is that here the data is arranged by 1 species in the pot to the most diverse, which is 3 species in the pot. And what we find is that sterile, that these are late sessional plants, they just don't grow. So if you put in the prairie mycorrhizal fungi, they grow, but it doesn't matter what diversity they are, so there's no over-yielding. But in post-agricultural soil, you do get benefits from diversity, and you get much stronger benefits with prairie soil community. So if you look at the actual composition is the mixtures of these 3 species, we can do it with these plots here. So this is sterile. Basically, this is biomass on this axis, different composition. This corner is all big blue stem. This corner is all the amorpha, and this corner is all the liatris. So with mycorrhizal fungi, big blue stem grows big. It doesn't matter. It's just basically overall yielding is in proportion to big blue stem. But with a more complex microbial community, you do get some curvature, and you get transgressive over-yielding in the native prairie soil. So in the most complex community, it matters. Total yield does depend on plant diversity in these long-lived perennial systems. So when we dissect the causes of that, we find that both accumulation of host-specific pathogens on big blue stem and improved efficiency of nitrogen fixation in the native system presumably due to the differences in rhizobial composition and mycorrhizal composition that contributed to this benefit. So if we look at the annual systems, what we know about the annual polycultures, intercropping is very common in China. About 30% of the land area of the crops are intercropped in many different combinations. And so we went to a long-standing experiment in Gansu province that was put in by Professor Li Long at the Chinese Agricultural University in Beijing. And so we started this in 2003 and looked at all combinations of wheat and maize and fava bean in rotation and in intercropping and in rotational intercropping. And so we went into this experiment and we looked at whether the microbial community contributed to over-yielding by assaying the growth response of these different species to the different intercropping or monoculture treatments. And we found evidence that changes in microbial composition, particularly pathogens, can be a major driver of the over-yielding that's observed in this experiment. They observed about 30% over-yielding, and that's what we were getting from the change in pathogen accumulation. So, in terms of a view of the potential management of the microbiome and what it can mean for perennial polycultures, I think we can be optimistic that inoculation with beneficial mycorrhizal fungi and rhizobia can improve perennial crop production. And it's quite likely that polyculture may be important to minimizing the problems of pathogens in these systems. And with that, I thank you for paying attention, and I'm happy to take questions. Yeah.
So, can you please use the mic for the recording?
I thought it was interesting that you saw, you saw a response in big blue stem, a positive response, but not so much in the Thinopyrum intermedium. And I'm thinking of two obvious differences between those grasses. Thinopyrum intermedium is a cool season grass. Big blue stem, I think, is a warm season grass.
Yep.
But also, the big blue stem is native. So, and you spoke about using native inoculums.
Yes.
And whereas intermedium is not native to. I'm not sure what you. Sometimes people use the word native rather loosely, but Thinopyrum intermedium is not from North America. So I don't know if you can speculate what is the difference in response? Why is there a difference in response between these two perennial grasses?
Right. I think that's a good question, and I think another difference between them might be in the longevity of them. Big blue stem might live a lot longer than intermediate wheatgrass, and I've talked to Lee about this. I'm not sure about that, but I guess another general result with the difference between C4, C3, I think there is a tendency for C4 grasses to be very responsive, but annual C4 grasses are not. So it really maps on better on the life history axis than it does to other correlates. And it's not perfect. There are long-lived plants that don't need mycorrhizal fungi. So I don't think we have the final word yet. On the native mycorrhizal fungi, you're right that we derive them from these prairies. But from what we've seen on lots of these types of comparisons, the biggest difference in overall responsiveness correlates with root architecture of the plant, and so it isn't so much dependent on whether the fungus is native, has co-evolved with the plant. For pathogens, it's very different. So the pathogens are very much depend on whether they're co-evolved, but the fungal response generally doesn't.
But you did
The mycorrhizal.
look at different species of. But you did see a difference in when you looked at different species of inoculum.
Yes.
You saw difference. There were some interactions I saw between species of inoculum and species of plants.
There definitely is, and a lot of that, but not all of it, is explained by generally good versus generally bad mycorrhizal fungi, but not all of it. You're right. There is some other specialization, specificity of response that goes beyond that, and so it is quite possible that different isolates of mycorrhizal fungi could really benefit Kernza.
Jim, great talk. And I mean, with those results, I'm surprised that the seed companies haven't already trademarked the AMF and are selling it already. But I was just wondering if you could speculate. Your results seem to indicate that the perennial plants that you tested responded more positively to the AMF than the annuals. Do you have any sense for the mechanism of that? Is it just increased time for the association to develop, or is it resource depletion, or what might be the mechanism?
So so these early sectional plants there's many selective pressures that they're specializing on. So they grow well in these disturbed soils, and so that involves having seeds that get to that site, but also they have to succeed where mycorrhizal fungal communities are disturbed. So mycorrhizal fungi will not reliably be in that environment. So part of their whole game is being successful whether mycorrhizal fungi are there or not. While these long-lived plants are always with mycorrhizal fungi. There's disturbed people. we refer to fire as a disturbance, but it's not a disturbance of the plant-microbe interaction. That's all static, so over long periods of time, these long-lived prairie plants always have their mycorrhizal fungi with them, so it's reliable. And so I think that's the difference, is whether you can evolve a dependence on a symbiont, knowing the symbiont's always going to be there, or are you going to evolve a strategy where you move into places where it may or may not be there. And then they differ in their investment in it. So other studies, we looked at the ability of the plant to allocate preferentially to the best symbiont, and these later successional prairie plants do that, and the early successional prairie plants don't. So they're just basically going with whoever's there, while the later successional species manage their microbiome.
An obvious question coming from me. Do you expect any changes in this IMF association with perennial plants when we start selecting for yield?
Yeah, we're very interested in that. So we're working with David with Silphium in testing the correlates of responsiveness to mycorrhizal fungi and yield in that species. So yeah,
what would be the mechanisms that you think that will be
well,
from the plant side?
Well, I mean, it could inadvertently come from selecting for yield in a post-agricultural environment. If you're in that system, it might be that the Silphium that doesn't invest in the fungus and instead puts more of its resources into acquisition directly through roots might be higher yielding. And so it could be a difference in yield with inoculation versus without. And that would be a really hard genetic trait to select on. But it could be important.
Well, as a soil ecologist myself, I enjoyed your presentation a lot. I've worked with alfalfa and rhizobia for a long time. And I found quite interesting your findings that the early succession plants didn't benefit that much as the late successional plants. I believe, or I think, maybe you agree, maybe you even found this out, that this has also to do with the nutrient content of this soil. Because all these successional stages or plants usually grow on, especially post-ag soils, on soils that contain a lot of nutrients, excess nutrients. And under those circumstances, mycorrhiza actually is not really necessary for mobilizing and for getting access to the nutrients. Whereas in late successional states, we're getting to kind of limitation of those nutrients. And that's when mycorrhiza comes back into the system and is needed for the plants to thrive.
Yeah, so I think that plays an important part. But some of these, it's commonly observed that the benefit from mycorrhizal fungi, for example, decreases as you add phosphorus. Most of those studies were done with early successional crop type plants. When you look at these really late successional prairie plants, they're locked in. They are totally dependent on mycorrhizal fungi. You can dump all the phosphorus on them you want, and they still won't really grow without mycorrhizal fungi. So it's sort of, so it could be that Kernza, so I think that you'd probably find more of that context dependence as you move towards the earlier successional plants, and that might be where Kernza is, that it could be that Kernza would really benefit in some soils and not in others. That's my reluctance with the Kernza greenhouse result, is that we found out later that was a really high phosphorus post-agricultural soil that we tested all those in. So it wasn't a fair trial.
Maybe the association between both plant and the mycorrhiza goes beyond the nutrients.
Yes.
Water, and we know
Yes, I think you're right, and they definitely can be.
Quite a lot of benefits.
Yeah, and a lot of plants are very dependent on mycorrhizal fungi in drought conditions. Yeah, you're right.